everybody, let's talk about Staphylococcus aureus epidermidis and saprophyticus, or Staphylococcus species. And this is going to be our first video in the microbiology series. Now, when we take bacteria can either be gram-positive or gram-negative, and this classification is based on gram staining. Typically, after gram staining, when the bacteria under the microscope looks blue or purple, we call it gram-positive, and this is so because it's able to retain the dye that is used in gram staining. That's the crystal violet, and so we have it called gram-positive. And then if the bacteria is not able to retain the crystal violet, it's called gram-negative and appears pinkish. But today we'll talk about gram-positive bacteria. Under the microscope, gram-positive bacteria can either look spherical or cylindrical, or like rods. When they are spherical, they're called cocci. When they are cylindrical, they are called bacillus. Our objective, today is to go through one of the gram-positive cocci bacteria, that is Staphylococcus. In another video, we'll talk about Streptococcus. Now, there are two main gram-positive bacteria. We have Staphylococcus and Streptococcus, and though they are both gram-positive and spherical, there are some differences between them. Microscopically, the Staphylococcus species appear in clusters. They are spherical and they appear like clusters, grape-like, under the microscope, whilst Streptococcus appear like chains under the microscope. But then to confirm, biochemically, we use a test called catalase test to differentiate between Streptococcus and Staphylococcus. And catalase is an enzyme that is contained by Staphylococcus species. It's actually a virulent factor. We'll get into it. We'll talk about it later in the video. But then we should know that both Staphylococcus and Streptococcus are gram-positive. That is, they retain the crystal violet. They are non-motile and non-spore forming and can cause similar diseases. So under the microscope, the Staphylococcus species look like clusters. They look, like this, like grapes, and then the streps look like they're in chains. All right. So talking about Staphylococcus, Staphylococcus species are gram-positive spheres arranged in irregular group-like clusters, and they are natural flora, natural skin flora. They are found on the skin, and also are facultative anaerobes. When we say an organism is a facultative anaerobe, it means it can survive in both oxygen and without oxygen, like that. So all staphylococcal species, or all staphylococci, produce the catalase enzyme, which is an important virulence factor, like I said. Now, in killing microbes, our immune system, typically the phagocytes, kill some of these bacteria using reactive oxygen species, right? And H2O2, or hydrogen peroxide, is one of them. Now, to escape the killing effect, staphylococcal species are able to produce catalase enzyme, which breaks down the H2O2 into water and oxygen with the reaction over here. So with the catalase, they are able to break down hydrogen peroxide, which is supposed to harm them, into water and oxygen gas, which is seen as bubbles, and then to escape the killing effect of the phagocytic cells. All right. So there are three species of Staphylococci which are important to humans. Like there are many, but then the ones that are clinically important or significant to humans are the ones highlighted over here. We have Staphylococcus aureus, Staphylococcus epidermidis, or albus, Staphylococcus epidermidis or albus, and Staphylococcus saprophyticus. Now, Staph aureus and epidermidis are natural skin flora. They are found on the skin.